Hi, my name is Emily Hu, and I will be presenting my topic on hereditary mutations in the keratin 9 gene, which leads to Palmoplantar keratoderma skin disorders. And my mentor is Ms. Jamie Moon of the UNF Biology Department. So to begin, I want to first explain what Palmoplantar keratoderma is, and just for future references, I'll be referring to this disorder as PPK. So what is PPK? It's a rare genetic autosomal disorder in which the skin growing on the palms and soles of one's hands and feet grows at an accelerated rate, thickening the skin. And this can be hereditary or acquired through one's lifetime. And there are four main types of hereditary PPK, which include diffuse, focal, stray, and punctuate types. So now that we know what PPK is and its classifications, we'll be going into some characteristics or symptoms exhibited by these individuals. So PPK is once again exhibited by a thickened yellow appearance on the skin of one's palms and soles. However, in some individuals, a red border can be seen outlining where the thickened skin ends, along with some of the keratotic lesions on hands, feet, knees, and elbows. And this is usually exhibited by the diffuse type PPK individuals. Other types have similar symptoms, but with a few distinctions. So, for example, focal PPK is seen along the pressure areas of the body, and striate is also characterized by fissuring, blistering, and sweating. In addition, formations of tiny bumps on the palms and soles can be seen in punctuate type, and in some cases, a thickened appearance of the nails can also be seen. However, this is not all, as even within the four types of PPK, various forms can exist within each category. So with so many types of PPK, I want to emphasize that the focus of this topic will be on the hereditary, autosomal dominant, diffuse type PPK. And this review will also focus on the underlying mechanism or functions of the keratin protein and its regulation by underlining the type and position of mutations in the keratin 9 gene. The statement or hypothesis that I will be supporting in this review is that diffuse type PPK disorders are caused by mutation in the keratin 9 gene, more specifically an amino acid missense mutation, which leads to abnormalities in the keratin protein, which controls expression and regulation of important skin factors. And just to re-emphasize, this review will only look at studies done on individuals with diffuse type PPK conditions. So first, some background information on the structure of keratin proteins. Keratin proteins are composed of intermediate filament proteins, which provide the shape and organization to cells. And these IF proteins comprise a central alpha helical rod domain of 300 amino acids with multiple segments flanked by 3 flexible non-helical linkers. And the helix initiation and termination motif, which makes up the 1A and 2B segments, comprise the start and end regions of the alpha helical rod. And these two regions are critical for keratin heterodimerization, in which obligate heterodimers make up the type 1 and type 2 keratin proteins. Therefore, mutations in the keratin 9 gene can be detected in these domains or near the regions. IF keratin proteins also contribute to many aspects of cellular behavior. Therefore, disruptions to the keratin filaments and organization of heterodimer molecule segments can result in these disease-causing skin pathology mutations such as BPK. Now, keratin proteins, which are expressed in the suprabasal cells of the epidermis, are regulated by the keratin 9 genes, and the role of these proteins is to reinforce the skin's response to external stimuli. So the keratin type 1 and type 2 proteins have highly specific tissue expression patterns, and the protein types are often expressed in specific pairs through a tissue differentiation program. And so these type 1 proteins are the special program that regulates the epidermal keratin of the soles and palms, and therefore mutations in the keratin 9 gene can affect the function and amount of protein type 1 on areas of the skin where PPK is usually seen. So like I had stated previously, keratin 9 gene mutations can result in abnormal keratin protein concentrations, either in type 1 or type 2. And from a study done, results show that abnormal protein concentrations can result in phenotypic differences that vary in severity despite being 60% similar in sequence due to alterations in the organization and assembly of keratin IF networks. And another study done by Hennies found that novel mutations in positions 156 and 171 cause mutations in the coil 1A segment, which is responsible for the heterodimerization of keratin and assembly of intermediate filaments. And this emphasizes an important concept that structure determines function. And since the coil 1A integrity is important for function, when the structure of the IF networks are affected, it's unable to regulate normal skin functions, and skin disorders such as PPK can result. So since I did a review, I just want to go over some of the recurring methods that was seen in all the papers I analyzed. So the affected individuals were usually compared to a group of control healthy, unaffected, randomized individuals from the population or their healthy family members. And PCR and gene sequencing were analyzed in these studies to determine the type of mutation, the exact position of the mutation in some studies, and what type of amino acid alterations were seen in these PPK individuals. In a study done by Shimomura, 5 Pakistani families with PPK were examined, and they found 2 novel mutations at positions 157 and 454, and 2 recurrent mutations at nucleotide positions 157 and 163 in the highly conserved helix motifs. And this supports that PPK is caused by keratin9 gene mutations, and that mechanisms in the hereditary formation of keratin plays an important factor in the cause of the disorder. So another study done by Race examined that keratin type 1 protein in the keratin9 gene found 3 mutations and isolated it to the human chromosome 17q21 and concluded that mutations also occurred in the highly conserved coil 1A domain. And in addition, the mutation at position 162 was identified across all 5 unrelated families, which affect residues in keratin 14 and keratin 10 genes, which can cause different forms of PPK. So these 2 studies provide support that underlying possible mechanisms of abnormal concentrations of type 1 and type 2 keratin protein exist, and that different mutations in their gene can cause different variations of the PPK disorder. In another study done by Fuchs Telem, the molecular basis of a Jewish patient was investigated for evidence for mutations in the keratin9 and keratin1 gene. So if you look at the figure on the right, you can see that there was a novel missense mutation at the keratin9 gene causing the diffuse type PPK disorder, and that amino acid valine was substituted for leucine at position 11, caused by a cryptic splicate activation in the keratin9 gene, which also deleted lines of amino acids coding for important functions during the transcription of the genes. The figure above was taken from a study done by Liu using similar methods to previous studies. As you can see in A and B of the figure, that these EPPK patients have a thickened yellow appearance on the palms and soles. In figure A, exhibits family 1 and figure B exhibits family 2. So PCR sequencing was done on these 2 families as shown in figure C, indicating that there was an amino acid transition from C to T at the nucleotide position 847 in the keratin9 gene. So figure D depicts the gel electrophoresis done on family 1 PCR results, where the 563 base pair band was present in the affected family members in lanes 7, 18, 21, and 7, while it was absent from the healthy family members exhibited by the lanes 8 and 19. So other studies looked at mutations identified in the exon of gene sequences, and a study done by Chen examined 6 affected Chinese participants with EPPK and a control group of healthy individuals. Blood samples and genomic DNA was analyzed, and PCR was performed to amplify the exon 1 of their genes to sequence for possible mutations. And the study found that there was a heterozygous missense mutation identified in site 488 of exon 1 keratin9 gene in the affected patients. And they found that a substitution of amino acid thymine for guanine and leucine for arginine occurred at position 163. Wang also conducted a study using whole exome and Sanger sequencing, and they found that mutations in the keratin9 and keratin 15 were causative gene mutations for PPK, along with a missense mutation at position 163 for arginine. So Western blotting and PCR was performed to analyze the effects of low mRNA and protein levels of keratin 9, and these studies confirmed that mutations were only seen in the affected PPK individuals, and that healthy individuals, whether family member related or unrelated, did not exhibit these mutations at the amino acid locations identified. In another study conducted by Cavello studying Northern Irish individuals with mutations in the keratin9 gene for type 1 keratin protein, a series of PCR sequencing was done, and it was identified that there was a heterozygous missense mutation in exon 1 of the keratin9 gene in all affected families, and it was found that there was a novel mutation in positions 158, 156, and 162, exemplified by the figure on the right, and a pyrimidine transition at positions 467 from thymine to cysteine was present for exon 1 keratin keratoderma gene, indicating that mutations in the position 156 of the helix 1A domain of the keratin protein causes PPK. Reverse strand sequencing and analysis of restriction enzymes was done to confirm, and figure B indicates that the NLA3 recognition sequence was disrupted by the 156 position mutation, confirming that the mutation in the proband of the affected family is shown by the upper uncut band in lane 3, which was not present in the normal individuals, indicating that a mutation was present. This is just a table I created compiling all the mutation positions for this disorder. As you can see, all the studies analyzed concluded that PPK results from a missense mutation in the keratin gene due to nucleotide or amino acid changes, which affects the 1A domain of the keratin protein, leading to diffuse type PPK. The purpose of this review was to identify the gene that causes the diffuse type EPPK disorder through studies examining the mechanism of the keratin gene, and it was hypothesized that the keratin gene mutations would result from amino acid alterations due to missense gene mutations which were identified, and it was also discovered that abnormal concentrations in type 1 and type 2 keratin proteins affect the heterodimerization 1A coil, which result in different forms of PPK. And mutations were frequently seen in positions 162 and 163, and it was also discovered that mutations in exon 1 can result in variations of the PPK disorder. However, since there were many types of amino acid substitutions and forms of mutations due to the complexity of the rare genetic disorder, this review poses certain limitations. Therefore, a complete evaluation and conclusion cannot be drawn, only that most mutations occur in the table presented previously. And furthermore, this review only examined the diffuse type PPK, which is only one of the 4 hereditary types found to exist. And since this disorder is very complex, more studies and research would need to be done to gain a better and overall understanding of the PPK disorder. At this point, I would like to thank Ms. Jamie Moon of the UNF Biology Department for her time and extraordinary guidance throughout the process of researching and writing my paper. And I would like to thank Dr. Casamata as well as the rest of the biology staff for teaching me all that I need to know to be successful during my time here at UNF. These are my references. And thank you for listening to my presentation.